Good afternoon, everyone. My name is Ken Cass, and I'm the Vice President of HD Care. We are welcoming you today to our educational symposium. This is the fifth educational symposium we have sponsored, but the first that we have done virtually. Previously, we have met on a Saturday morning at the Sue and Bill Gross Stem Cell Research Center on the campus of UC Irvine. In addition to being a great meeting environment, the Research Center is so appropriate since it is the home for much of UCI's Huntington's disease research. The pandemic made an in-person gathering infeasible this year, but we didn't just want to bag the symposium for the year. There is so much going on that we wanted you to hear about. We decided to use the technology that's empowering this country's business and academic enterprises. So, welcome to Zoom and the HD Care Virtual Educational Symposium. While this format was born out of necessity, we think it provides flexibility to make the symposium universal. Many people can attend who have not been able in the past. For instance, allowing family of the local HD community to attend from across the country. We really have no boundaries, either physical or time. It also allows us to engage HD experts who couldn't have invested the time for a 3-day trip to Orange County. As you will see, we have virtually assembled a really diverse and expert group of speakers. All we are missing are the fresh fruit and bagel breakfasts and the bagged lunches from days of yore. We're eager to see how all of this works out, and we solicit your feedback at the end of the symposium. As in years past, we have tried to set an agenda that will provide information and we think encouragement to all the members of the Orange County HD community. The structure of the symposium will be much like in previous years, only we'll be doing it remotely. We are again fortunate to have Dr. Peter Donovan as our guide through this afternoon's interesting agenda. We strongly encourage people to submit questions and comments during the presentations. You'll see a Q&A button/box at the bottom of your screen. You can use that to type in questions. Peter will keep track of the information flow and consolidate the questions and present them to the presenters at the end of their presentations. Because the symposium is scheduled for 2 hours, and because we have many germane topics, please understand if not every question gets answered today, this afternoon. We will attempt to answer all the questions and post those on the HD Care website very shortly after the symposium ends. Before I turn the screen over to Dr. Donovan, I'd like to pass along an anecdote that explains why HD Care exists and why we think this kind of symposium is so valuable. Twice in the last several months, people have sent messages through the HD Care website saying that they've learned that there is HD in their family and asking how they can learn more about HD and how to be tested. HD Care is one tile in the mosaic that represents the lives and activities of the Orange County HD community. So when you donate to HD Care, you are supporting research, you are providing education, and you're reaching out a hand to people who are trying to find their way. That is a really wonderful, wonderful benefit. I now want to turn the symposium over to Dr. Peter Donovan.
Thank you so much, Ken, for those introductory words. And I think we're going to have a great day today, a great couple of hours. And as you said, in the world of Zoom, we've managed to get some terrific speakers from the other side of the country and from Canada. And maybe that's something would have been difficult without this amazing technology. So to all of you, welcome. We're really glad that you've decided to join us today and join the UC HD Care community, which has really grown since its inception several years ago. And it has been lovely over the last few years to have seen this symposium grow from a few people to filling the auditorium at Gross Hall. And so we're really pleased that we're able to continue it this year by inviting guests from all over the place and to share the amazing research that's going on and the spirit of hope there is to be able to develop new treatments for Huntington's disease. So the next two speakers, I'm very pleased to introduce. Mitra Hushmand is one of UCI's own. She did part of her training in this building here, Gross Hall, which I'm sitting in, and she did her studies here, working in the lab of a noted spinal cord injury scientist, Eileen Anderson, and she won a number of awards. And then after graduating, she went to become Director of Scientific Programs for Americans for Cures, the organization that is really promoting the work of regenerative medicine, not only in California, but around the world. And it's a delight to be able to introduce her. And she will present with another person we've known for a long time, Melissa King, who is a very talented individual who worked initially with the chair of the California Institute of Regenerative Medicine when the California Institute of Regenerative Medicine was first started. And now she's currently serving as the executive director for Americans for Cures and is really pushing towards getting a new initiative in California, which will be really building on this fantastic initial initiative, which has been groundbreaking in its scope and the work it's accomplished. So welcome, Mitra and Melissa, and thank you for being with us today.
Thank you so much, Dr. Donovan. It is always a great honor to be with you and the HD Care Group. We can't say enough wonderful things about the work that Dr. Thompson and you and your colleagues at UC Irvine do, including in the lab where Mitra got her PhD. That's one of my favorite stories to tell about having been involved in this effort for as long as I have, which is that I've been around from the beginning working on Prop 71 in 2004 that I'll tell you all a little bit about. And now I have an incredible colleague in Dr. Mitra Hushmand, who got her PhD in a lab, in a building, all funded by an agency that I was part of helping bring into existence. So again, thank you so much for having us here. One of our heroes, Frances Aldana, is on the first slide that we have there. And I want to talk to you today about how people like her, when they come together in a broad coalition, can do even more that they do as individuals and with individual organizations. And you all know how much Frances does and HD Care does. So let me tell you a little bit about what happens when those of you in groups like that come together. In 2004, we had a ballot initiative on the ballot for the state of California to allot $3 billion to stem cell and related research with the plan of it lasting about 10 years, given it was a new field and we needed to fund some basic research and some translational research. There was not a lot of clinical work going on at that time, certainly not any human clinical trials, because the field really did need to evolve, which was part of our reason for existing. And we got that initiative passed with 59% of the vote, which is a wonderful endorsement. And at that time, we had 80 patient advocacy organizations that endorsed the initiative and also helped us get the word out about it in much the way that HD Care and 90 other advocacy organizations are helping us do this time around. Next slide, please, or thank you. So what resulted from us getting Proposition 71 passed in 2004 was the creation of our very own California State Funding Agency, similar to a mini NIH in California, but focused on stem cell and related research, because this was a new field at the time, still relatively new. Embryonic stem cells had only fairly recently been isolated and brought into the tool set of amazing researchers like Dr. Thompson and Dr. Donovan. And at the time, there were some ideological reasons that it was being kept from being funded at the federal level. So we needed to fund it at the state level, especially here in California with all of the world-class institutions that we have and an economy that is as big as most countries in the world. We're either the fifth or the sixth biggest economy in the world, and we have more world-class institutions than most countries. So we wanted to fund this research at the state level to build a foundation that brings us to where we are today. Next slide, please. And what has the impact been of having this institute, the California Institute for Regenerative Medicine here in California? Well, as I've talked about, the field has evolved, and now we have actually more than 60, it changes every day, it seems like, more than 60 human clinical trials directly funded by CIRM. And in addition to that, another 30 or more for which the earlier stage research was funded by CIRM. And you'll hear a little bit more later from Dr. Hushmand about how that's impacted UC Irvine and specifically the Huntington's disease community as well. 4 of the CIRM-funded clinical trials are in phase 3. That means that while no one can make any promises either about the end results or the timeline of science, we're getting close. We have some very good shots on goal right now, and we're very hopeful. Over the past 15 years of funding during which CIRM has been able to fund research, there are more than 2,900 peer-reviewed publications in the top journals. So these are scientific projects that were carried through to being able to be published by world-class peers, reviewing them and placing them in the top publications in the world. We have, wow, that's a big number, more than 2,200 training grant alums funded by CIRM that now are, including medical residents and fellows, that provided, this training provided additional education for people that had already decided they wanted to be MDs. They wanted to practice medicine, but they didn't know how to use cellular therapies. And this provided the training for them to do so, so they can participate in these clinical trials that now exist. And oh, this is wonderful. One of my favorite things to talk about from CIRM and from my 6.5 years working there, helping build the agency, it was the patient advocates on the board of CIRM in the earliest days of the agency that said, we absolutely want funding to go to the world-class institutions and scientists like UC Irvine and Dr. Thompson and Dr. Donovan, but we also want, especially since this is state funding, for the funding to be provided to a broader set of Californians. We want to get it to the people at the state universities, the CSU campuses, and the city colleges. And one reason for that is to try and grow the workforce for the development of the treatments and cures that we all hope eventually will result from funding this research. But really what's happened with it is a bunch of people have come through the program, and some do go into the biotech industry, and that's wonderful, but many end up themselves going on to get PhDs or MDs or both, and are adding to the ultimate lexicon of what we need to know about stem cells and the research to bring the treatments and cures to fruition. 80% of the alums from that program do advance to graduate school or full-time employment. So by any measure, it's been a success. Next slide, please. Oh, yes, 2 FDA-approved drugs in blood cancer. So CIRM-funded research led to the development of 2 blood cancer drugs. This work was done at UC San Diego by someone funded in the very earliest days of CIRM with a new faculty grant, and her amazing career has been built partially on the back of that grant from CIRM. And she is keeping people with 2 forms of blood cancer alive with 2 drugs that have gone all the way through the FDA approval process. And her earliest research all the way through to when she was ready to put those drugs into clinical trials was funded by CIRM. Thank you. And at this point, I'd love to have Mitra talk you through some of the amazing successes on the scientific level of CIRM. Thank you.
Thank you, Melissa. So I'll go through these quickly so that we can leave a lot of time for question and answers. Just wanted to cover a few of the broad application discoveries conducted by SERM, which will hopefully apply to a variety of different disease settings. The first of which is the development of approaches for enhancing immune tolerance, and this will hopefully allow for patients to receive stem cell transplantations without aggressive chemotherapy and radiation. There are imaging techniques that have been developed as a result of SARM funding for tracking cells after transplantation, which is incredibly important from the perspective of understanding what the cells are doing once they're given to the patient. The world's largest iPSC repository has been made with the help of SERM funding, and this is essentially, you can think of it as a stem cell bank that holds on to cells from a variety of different patients, including HD patients, in order to better understand the manifestation of the disease. And lastly, for the purpose of this particular slide, the establishment of centers of excellence, which are incredibly important for making sure that patients have access to evidence-based clinical trials. And you may have heard of these as alpha clinics. There are 5 of them. One is located, in fact, at UCI. And the purpose behind these alpha clinics is so that they can give access to patients to legitimate clinical trials and help patients along the way as they get exposed to the research behind the clinical trial and what the trial itself entails. I wanted to just take a minute to also cover the impact of SERM specifically to the HD community. There has been $35 million that's been given in total to HD research. 7 of those grants have been awarded to the rock star of this group, Dr. Leslie Thompson, who is really spearheading a lot of this area of research in Huntington's disease. And you may know that most recently, one of the grants that she was awarded was to facilitate IND filing, which will hopefully enable her to soon get into a clinical trial using human embryonic stem cell derived cells for HD patients. So we're all very excited to see the results of that. But I also wanted to mention that 23% of SERM funding has been awarded to neurological diseases. And the reason why this is important to indicate is that there's a lot of overlap when it comes to CNS, and understanding of one disease can really change the way that we think about, understand, and even try to treat other diseases. And so as a result of the totality of this funding, the field is really moving forward significantly. And that brings me to another really key impact of SERM, and that is the bridging of funding. As Melissa indicated at the very beginning of this talk, clinical trials are difficult to conduct, they're costly, and oftentimes going from one area of science to another, as you can see here depicted by basic science, which is the kind of science we're doing in a lab as we're looking under the microscope at some cells, going to clinical trials and all the way out into clinical practice, which is when cell therapies or any other drugs are available to the market, has a lot of potential pitfalls. And these are sort of these valleys of death that the therapies could fall into and never get out of. And a big part of the reason why this happens is because of lack of funding. And what SERM has done is try to bridge this gap by funding some of the early studies that the biotech industry perhaps doesn't want to take a risk on. And they come in, they de-risk it, and allow for that therapy to continue and make its way out into clinical practice. And finally, I just wanted to cover the impact of SERM on economy. The forecasted impact is fantastic, and the California impact that it's had so far, with 55,000 jobs, full-time equivalent jobs that have been created, lots of money in terms of gross outputs and tax revenue, not just within the state of California, but for the entire nation. And you can imagine, obviously, with this increase in job creation and the ability of many of these therapies ultimately to get out into clinical practice, not only are we saving, not only are we making jobs and generating revenue, but we're also saving a lot of money on healthcare potentially. So with that, I'll turn it back over to Melissa to tell you a little bit about how we can continue this kind of funding.
Thank you so much. And I will be as brief as possible. I know we want to leave a chance for you to ask us some questions right after this presentation, or if you don't think of it then, you can ask them all the way at the end of the symposium as well. But so how can we continue all of the wonderful work and the necessary and stable funding that has been provided for that work by CIRM over the past 15 years? Well, we do have an opportunity this November 3rd, really over the next few weeks, people are voting early, to consider another ballot initiative. Proposition 14 is on the California ballot, on the November 3rd ballot, and it would authorize $5.5 billion in state bond funding for the research. And that number is a little bigger than the $3 billion of 15 years ago. And the reason for that is because, as Mitra and I have both been talking about, we're now in clinical trials, and those absolutely cost a lot more than basic research, but we also have to go back, continue funding basic research, funding the translational research that leads to the clinical studies. And in order to do that, we need to have a larger number. So the initiative for $5.5 billion specifically allots $1.5 billion to the brain-related, the central nervous system diseases, as Mitra mentioned. And again, the reason that's important, first of all, is because these are such tough diseases to figure out, and we're so lucky to have people like Dr. Thompson working on them. But also because, as she said, there's a lot of overlap, and research into one disease may lead to something that would be helpful for another, and that's exactly what we want to see. I can tell you that we have, like I said, 90 or more organizations at this point. That number increases every day that are supporting the initiative. You're seeing the logos of those, many of those on the screen right now. And just encourage you to consider supporting Prop 14. You can check out their social media feeds that are on the slide in front of you. And we'd love your help. If you are supporting Prop 14, we'd love your help getting the word out about that between now and November 3rd, so that we can continue this stable funding at the state level, which is the best way to fund this research that we know of in California, and all this world-class research like that happening at UC Irvine. Thank you.
Thank you so much, Mitra and Melissa. And just for our audience, let me say, if you want to ask questions, please pop them in the Q&A section, which is down at the bottom of the Zoom screen. If you don't mind, I'd like to ask you guys a question that I think a lot of people might ask, which is, with this pandemic going on, you know, why would we want to be spending money on stem cell research? What has stem cell research got to do with COVID-19? And what is CIRM doing about that?
So I'll start. Thank you, Dr. Donovan. And I definitely want to have Dr. Hushmand address the scientific reasons. But one thing I'll say is we're in the middle of a pandemic, and we're also experiencing a challenging economy related to that. And the great news about Prop 14 is that while it is asking the state to spend money, yes, it's $5.5 billion in bonds, and ultimately we as Californians will pay for that. But it is amortized over about 40 years, and zero payments are needed or required from the state for the first 5 years after the initiative is passed. And we wrote it that way specifically so that the costs of it are amortized over more than one generation, because more than one generation will benefit from the results of the research. So that's the first thing. And the second thing is you can actually consider this because of the fact that no payments will be made by the state for the bonds used, the bond funds used for this initiative, this research that will be funded through CIRM. This is actually an economic stimulus program right when we need it the most, right when our economy is going through some challenges. So that's the financial piece of this, and Dr. Hushman knows a lot more about the science than I do, so I'm going to have her talk about that. Thank you.
I just wanted to add one point about the science behind this is that STRM actually within weeks after the COVID pandemic put together an emergency meeting and allocated an additional $5 million of funding specifically to COVID-19 research. And it has already funded 3 clinical trials directly looking at how we can use stem cell therapy as a means to combat COVID-19, as well as multiple basic level or discovery stage grants. So it's actually immediately tied into the current state of the pandemic.
And I think, Mitra, just to expand on that, because stem cells offer the opportunity to make many cell types in the human body, potentially SERM could address issues to do with the biology of COVID-19. For example, one of the issues that's relevant to this group, I think, is what's the impact of COVID-19 in the brain, and what would happen if a Huntington's patient got COVID-19?
Absolutely. And what I found really interesting most recently is that, in fact, some of the studies that are done using brain organoids, which I'm assuming somebody may even be talking about today, which are made using pluripotent stem cells, have been able to tell us a lot about what's happening when brain cells are exposed to COVID-19.
And of course, I think as well, because we can make every cell type, it's possible to presumably make airway epithelial cells and study how the virus infects those in the first place.
Which absolutely is being done. In fact, I think several of the discovery grants are related specifically to that.
Wow. And you both talked about training opportunities. Melissa, why was there the need to train people? Why couldn't SERM have just spent all the money on research up front? And what's the benefit in the long run of that training?
Sure, it's a great program. So it's a great question, excuse me. And one of the first programs that SERM funded, going all the way back to 2005, was a training program in stem cell research for graduate students at various campuses, including UC Irvine, as I'm sure you remember. And the reason that that was important was because the field was new. And in order for people to actually pursue stem cell research as a career, as their area of interest, they needed to be trained in a concentrated way how to work with those cells, because it was so new. It was such a new opportunity. The other types of training that we've talked about, the Bridges program, encouraging people at CSU campuses and city colleges to get involved in science in a way that their own campus wouldn't entirely allow them to. So it brings them to a lab on a campus like yours. Those are important for so many reasons, whether it's equity in the funding and how accessible it is to all Californians, but also inspiring additional scientific careers. And then the other type of training that we mentioned is training for medical doctors, that they've already gone through medical school and they're already working clinicians, but they do not know how to handle cellular therapies. So researchers like you and Dr. Thompson, if you need people to help you conduct your clinical trial, they don't know how. We all think doctors are amazing and brilliant because they are, but that doesn't mean they know everything. And if there isn't concentrated funding to train them for this, there's not really a way for them to be trained. I'll leave it there. I hope that addresses your question.
And I do want to keep on time, but there is a new question on the Q&A that I think is worth asking. And this is from June Ann Gold. Who is collecting the data on those who've had HD and COVID-19? And I'm not sure that's you are the right people to answer that question, but I think that any medical center that has a patient with COVID-19 collects that data and reports it to the local state agency. Presumably in the medical chart, that data would be recorded, and then people could go back retrospectively with the benefit of privacy, the HIPAA regulations, and identify patients that have had both COVID-19 and HD.
Dr. Donovan, I just wanted to address, there was one other question in the Q&A earlier from David Saldana about the the economic impacts of Prop 14 and where that information is. There is some, as he asked about, in the voter guide. There's a little bit of that in our argument in favor of Prop 14. There's a lot more of that on our website at ca4cures.com. And another way you can get that information, you can always contact Mitra or me, and we are happy to point you to as much in-depth information as you need.
Thank you very much, Melissa and Mitra and Junan. I hope we answered your question, but as Ken said at the beginning, we'll go back through the questions and post as much as we can on the website. So thank you very much.
Thank
and now
you.
It's a pleasure to
Thank you.
thanks, guys. Yeah. To introduce the next speaker, Melody Bandley. And you'll notice on the program, it says Melody Bandley Esquire, which means that Melody is a lawyer. And she qualified, she passed the California bar a few years ago and is a practicing lawyer, and is one of these people that it's always been a pleasure and an honor to get to know, which is someone whose relative developed HD and has become immersed in the cause and has helped the HD Care group quite a lot. So Melody, welcome, and thank you for joining us this morning.
Well, thank you. Thank you, Peter, and thank you for inviting me to participate in this symposium. My family's journey with HD began 6 years ago. We are not a family that has had it lurking in the background where we knew it was a possibility. I had never heard of it. And my husband started having some tick-like symptoms, so we went to see a doctor and were referred to a neurologist. The neurologist kind of said, like, well, heaven forbid, maybe we should test for Huntington's, but we couldn't get authorization because there is no history of it in our family. But eventually we saw Dr. Hermanowitz at UCI and he was able to get authorization. We had the test and turned out he had Huntington's. So that was quite stunning and a real change to our world. Initially, my husband was having very minor symptoms, and his symptoms progressed very slowly for the first, I would say, 4 years or so. He didn't even notice them. He only went to the doctor because others suggested he do so. But when we got the results, initially he didn't want to tell anyone, not even his mom. But we did soon tell family, and sooner than I thought he would, he told his boss at work. I was kind of worried about that, but that turned out actually in our case to be a blessing because he was able to work for 6 more years. He's on leave now recently and is preparing for retirement, but he could not have worked as long as he did without them understanding that he had HD and trying to support him. So I'm grateful for that. Also, when he got HD and I started thinking, of course, then I worry about my kids. We have 3 children. One of my daughters, well, I have 3 children, 1 daughter. My daughter decided fairly early on she wanted to be tested. And so almost 2 years ago, she was tested and she does carry the gene. So that was more stunning, more upsetting, more everything than my husband's diagnosis. I could handle my taking care of him and all of that, but to have my children, that's another story. Sorry. So that really made me kind of, okay, I need to up my game. What can I do? I need to fight this more than I have been. I started working a little more with Frances Saldana and those at HD Care. I met Frances at one of the HDSA conventions that I attended, and she is literally a force for good in the HD world. She has done so much, of course, founding HD Care itself, and advocating for so many good things, and she inspired me to take more action. Also at an HDSA convention, I saw a presentation by Paul Dennis, which has stayed with me ever since. And I'm thrilled that Paul Dennis will be presenting later on today in the symposium. And that is one good thing that's come from COVID, is that, as Peter mentioned, we're able to have those that would not be able to be here otherwise participate. And I have wanted to have Paul present since I saw his presentation several years ago. And I'm looking forward to hopefully next year being able to have him actually do a class for those with HD to show how his work actually works. But I know that one of the reasons that I was asked to participate is because my husband is in the Roche Genentech study currently underway for Huntington's disease. And I know there are those that are kind of probably wondering, what does that entail? Well, it entails a lot. I'll just give you a quick summary. The first visits are, well, you have to go 3 visits in quick succession. For us, we have to go to Pasadena. And they do an MRI, lab tests, a large number of neuro tests, and interviews. And they're, once he qualified, then they do the first lumbar puncture, which necessitates him holding still. I'm sure they help him significantly. And they insert a large needle in his spine, draw out a set amount of fluid, inject the same amount of study drug into his spine. And then he walks around for a few minutes and we go home. So the first time they did this, then the needle broke off in him. That was really exciting. And so they had to do it twice. That was an equipment malfunction that has not repeated. The second time he had it, he got a huge headache and was very sick. He was throwing up for a few days. It was of some concern. But good news, since then, he's had several, and the last one, he didn't even take Advil after. He just went back to his activities. And so it appears that either they're getting better at doing them or he's getting better at tolerating them. I don't know. But at any rate, that's the scary part of the study, is the lumbar punctures. And he takes them like a champ. And now we don't even dread those. He does also wear a smartwatch and a device that looks like a cell phone every day. And they monitor literally his every movement. And then he has daily tests that he does on his device, where he does movements, where he actually speaks to it and does neurologic testing, kind of similar to what he gets in the clinic. And then I have my own device, and I have tests that I fill out every week, once a week. The lumbar punctures are now every other month, and bless them for changing that protocol. So we don't have to go every month. It really makes a difference to have that month off. And also we get an MRI every like 6 months. So, and initially when my husband went to the first 3 appointments, he's like, okay, I'm done with this. But the good thing about that Huntington's disease OCD stuff is once you get past that first routine shock and he gets it in his system, well, it's just, I don't even remind him about his test. He just does that. And we just like, when do I go next? We're in that, routine now. So, that's kind of the study situation. It's 2 years long. So this will continue for us until next June. And you're probably wanting to hear if I think that he's doing better when he's on the study. Well, I won't give you that information. You can stay tuned. I think we're in the placebo group, but who knows? We'll see. Anyway, I kind of think that's all I have prepared to share with you. I don't know.
Melody, there's a
Yeah.
question in the question and answer section that maybe you could help address, and that's from Hervig Lang. And that is, he asks, how come you need authorization to get a diagnostic test?
Well, That's a good question. You don't need authorization if you have $1,000 to pop down to pay for the test. But if you don't, then you need authorization from your insurance company. Now, when Dr. Hermanowitz explained my husband's symptoms and that he'd had all these other tests, because the first neuro did all these other blood tests and there was no other explanation. Honestly, my daughter, who knew nothing about Huntington's at the time, diagnosed my husband before the doctors did. She was like, I know he's got Huntington's. Great. But anyway, so yeah, once we got them to explain why he needed the test, then it was paid for and we did it. We didn't want to pay on our own.
Now, people probably think that all lawyers are fabulously wealthy, but there's an aspect of Huntington's disease that I've realized over the years, meeting families, that it puts an enormous financial burden on families. After diagnosis, how long was your husband able to work, and what was the financial impact for your family?
So we have been very blessed. My husband was able to work for 6 years. Honestly, on the side, I called Dr. Hermanowitz myself and said, hey, how long is he going to be able to work? He said 2 years. And I was just like,
Yeah.
At the time, I mean, I still have 3 kids that are unmarried. They're grown, technically, adults. But, you know, they're still my kids, and I still feel, you know, most of them are still in school, that kind of thing. So he was able to continue work, no impact financially. However, he is retiring this year. And yes, while we're also blessed with a good retirement because he worked at a good job for 30 years, the financial ramifications are about $100,000 a year. I mean, it's very significant. And I am an attorney, but I don't work. I haven't worked in a number of years.
Yeah.
So my kind of thing is, do I go back to work? I can't now.
Yeah.
So, yeah.
So one of the things that must concern you as a parent, because no matter how old your child is, they're always your baby. You said your daughter was diagnosed. You must be concerned about financial things for her going forward.
Yes, yes. We have a lot of hope that things will work out with some of these marvelous clinical trials. And also, that's the advantage of her knowing early on, I feel. And honestly, we wanted her to be tested because we were so sure she would be negative. I mean, that's not the whole bit behind it, but we really felt and knew she needed to be tested. Now we feel that's so that she can be an advocate, that she can be prepared, and she will be. She'll be so much differently prepared than my husband, who just like, okay, this is something we ignore. He still ignores it.
Yeah.
You know, he just goes about as much as he can and pretends it doesn't happen. But my daughter is like every day, I'm working on this, I'm working on this, I'm working on this. And so it's strengthened our family, and especially the bond with my daughter and I, and my daughter and her husband, immensely. I do see a question that I want to respond to about the clinical trial.
Yeah.
When my husband said, no, I'm not doing this, I said, yes, you are. And I said, you know, there are people all over the world that would give anything to be in this trial.
Yes.
And we've got 3 kids. At the time he joined the study, did we know? No, I don't. The time kind of, I'm not sure if we even knew that Jenny was positive at the time. I don't think we knew. And I'm like, you have to do it. They need people to find out if this is going to be helpful. And even if you don't if you get the study drug, great. If you don't, well, then you're still helping. And after I did that, he didn't complain anymore. He just sucked it up and did it. So, yeah.
Melody, thank you ever so much. I think, let me say on behalf of the scientists, hearing the stories of how families deal with it makes it much easier and worthwhile to go into the lab and keep working on this, because I think the families often think, oh, isn't that great, the scientists doing this work, they're our heroes. But let me tell you, it's usually more the other way around. We really admire the way that you guys deal with it, and it makes it, you know, worthwhile going into lab every day to help try and fight this awful disease. So,
Well, thank you. I have to say vice versa. I mean, we pray for you guys all the time. And really, I feel so much like just that the sign, I'm so impressed and amazed and thrilled by all those of so many different walks, and especially the scientists that make those efforts to find cures, to find new ways. And I appreciate it. So thank you.
Melody, there's one quick question that came in. Can participants use supplements to boost brain repair? I know that you're not necessarily up with all the clinical trials, but anecdotally, do you have a thought about that?
Brain repair? No, I don't have anything that I you know, have heard or know or anything like that. I will anecdotally say, when my husband started taking melatonin, he takes it every day, 5 milligrams. His sleep became more calm. He sleeps very well, which is, as many of you know, it's a struggle with people with HD. But he sleeps very well. And I used to notice more disturbance in his sleep, but he sleeps much more calmly since he started taking melatonin. So I think that that's a helpful thing. So
Okay.
that's my 2 bits.
Thank you ever so much. I really appreciate it. Yeah, thank you. So now we move on again now to a little bit of research. And again, it's an enormous pleasure to welcome the next speaker, because I know Sarah very well, because she works here at UCI in the laboratory of my colleague and the rock star, as Mitra said, of the HD research here at UCI, Leslie Thompson. And I've known Sarah for quite a while, and she's passionate about her research. And she also has played an important role in working on the board of HD Care to kind of push the other aspects of the mission, which is to build this community of researchers, patients, patient advocates, and fundraisers to really drive research in this area forward. So Sarah, thank you for joining us this morning, and look forward to hearing what you're going to talk about.
Thank you, Peter. So this talk is going to be a bit different for me because it's not actually about my own research, but I'm hoping it will be a fun way for everyone to learn more about recent scientific findings related to Huntington's and how popular press articles may be over-interpreting that data that might cause them to publish what can come across as misleading headlines. And so these are the type of headlines that really make your eyes pop out when you read them and you think to yourself, can this really be true? Maybe they cured Huntington's. So we'll go over 2 headlines today, and then I'll break down the real science that was found in those primary articles that the headlines were referring to. So this is our first headline. It came out in June of 2020, and the headline is, "Antihistamines could slow progression of Huntington's disease. Researchers say the drug could help ease patients' symptoms at the earliest stages." So if you're anything like me and you see a headline like this, you immediately want to run out to CVS and buy some Benadryl or Claritin because maybe these antihistamines could slow progression of HD. But is this true? Unfortunately, no, this is not true. But the authors of the paper that this headline is about were using an antihistamine in Huntington's research. So let's go over specifically what that paper looked at. Histamine is a chemical messenger called a neurotransmitter, and you may have heard of other neurotransmitters. So these are things like serotonin that make you feel happy, or adrenaline that give you that rush. And each of these neurotransmitters causes these reactions by binding to a receptor in your brain, and the same way that Legos interact. So it's the connection between the neurotransmitter and their receptor that causes the effect that these chemicals are known for. So when serotonin binds its receptor, it gives off that happy feeling. Dopamine is a neurotransmitter that is responsible for the reward signal. So it creates that feel-good reaction, like when you win $10 on a scratch ticket, or you get a good grade on a test that you studied hard for. But dopamine also plays a major role in Huntington's disease because it helps to control movement. So this is a section from a brain. And when people die, they can generously donate their brains to science, whether they have Huntington's or not. And scientists can then take a thin section of that brain and study it for various purposes. So if we're looking at this brain and it was inside a head, the person's ear would be over here to the left. So normally there are certain brain cells that put out dopamine to tell the body when to move. And there are other specific brain cells in an area of the brain called the striatum, which is this bubble looking thing here, that receive that signal to tell the body to stop moving. And so this allows the body to really balance movement using a single signal from a single molecule. In Huntington's, the area of the brain called the striatum shrinks over time because those cells are eventually lost. And additionally, the cells that put out the dopamine signal to tell the body to move put out way more than they should. And when that signal is when the cells in the striatum try to receive that signal, there are fewer cells there, so they can't tell the body when to stop moving. And so this causes something called dopamine overload, and it makes the movement reaction out of balance. And that dopamine overload can further harm cells in the brain. But there are drugs that maybe people in this audience have heard of, like tetrabenazine or Haldol, that specifically target that dopamine reaction and try to restore balance. But those drugs that act directly on dopamine can have various side effects. So researchers have been trying to find other ways that they can reduce the dopamine overload without directly targeting dopamine. And that's where antihistamines come in, because dopamine also interacts with histamine in something called a hybrid receptor. And normally in a Huntington's brain, when dopamine binds that hybrid receptor with histamines, this can contribute to dopamine overload. But if you add something to that receptor to block dopamine from binding, the signal can't get through. So dopamine is still present, as is histamine, it just can't bind that receptor. And so this is similar to if you've ever used a landline and someone's already on the other end, the signal won't go through. It's the same situation here. The antihistamine is blocking the dopamine signal from going through. So by blocking these signals, the researchers can restore balance without actually reducing the amount of dopamine present. But this brings us back to our original question: should you start taking antihistamines to reduce HD symptoms? The answer is still no, because the antihistamines that the researchers used in this study aren't the kind that we typically think of that control things like sneezes and watery eyes. There are actually several different classes of histamines, and the ones that most people know are H1 and H2 histamines. So these control things like seasonal allergies or even heartburn. And we have very reliable drugs that can block H1 histamines like Benadryl and Claritin. And we also have very reliable antihistamines for H2, like Zantac and Pepcid AC. But the histamines that the researchers in this paper looked at were H3 histamines. And this was the type of histamine that they tried to block. And they did this with an antihistamine called thioperamide. And this is a very specific antihistamine against H3 histamines. And so now we can add to our little Lego cartoon. So we have dopamine binding the hybrid receptor specifically with H3 histamines. And we know that in a Huntington's brain, when the dopamine binds this receptor, it contributes to dopamine overload. So by adding thioperamide, we still have dopamine present, so we won't get any of those side effects from drugs like tetrabenazine or Haldol, but the researchers were able to restore balance. But again, you could ask, so should you start taking thioperamide? And the answer is still no, because this was more of a proof of concept study to show that dopamine signaling can be blocked in ways that don't directly target dopamine, and that instead target hybrid receptors. And so far, this has only been done in mice, but future studies are very likely to explore drugs that take this approach of trying to block hybrid receptors. So if we go back to our original headline, we can now break down exactly what this means, and hopefully we understand it a bit better. So did the authors of the paper actually use antihistamines to slow Huntington's, and can it be done? Well, maybe, but certainly not Benadryl, and certainly not the kinds that we typically think of. And can this type of research ease patient symptoms and balance the dopamine overload we see in the brain? It's currently only been done in a mouse model. So basically this headline was definitely misleading. And while this research was really interesting science, the headline doesn't quite capture what the researcher showed. Moving to the second headline, this came out in October of 2019, and it says, Huntington's disease genetic mutations expand throughout life. So if you're like me, when you read something like this, you might think, okay, if I get my HD test when I'm 20 and I have 42 CAGs, maybe if I get tested again when I'm 40, I'll have 65. And by the time I'm 60, I could have 109. Or alternatively, if I test negative for Huntington's and have 19 CAGs when I'm 20, could I have 45 CAGs when I'm 60 and actually develop Huntington's? No, that is not at all what was shown in this paper. So let's look at what the researchers actually found. We tend to think of DNA as a perfect blueprint to code for who we are as a person, but DNA is constantly being used. It's being read by various molecules that create the proteins that make up our bodies. And like all things that get used a lot, it has to be maintained and repaired so that mistakes don't happen. And the proteins that help repair DNA are very aptly named DNA repair proteins. But sometimes repairs can go wrong. So if these proteins were meant to repair this blue part of the DNA, they might have added an orange part. And so these mistakes tend to happen when there are long repetitive sequences of DNA. These tend to cause molecular mistakes or molecular stutters. And this is like the difference between copying these two pictures. One is very simple and one is incredibly detailed. And if I asked you to try to make a perfect copy of both of these, you would obviously have a much more difficult time trying to copy the photo on the right because it's incredibly detailed. And the same is true for DNA. The more detailed the DNA is, the more difficult it is to repair. This is the sequence of DNA for Huntington with 42 CAG repeats, which I've highlighted here at the top in yellow. And the longer and more complex the gene is, the harder it is to repair. And one of the things that makes genes very complex are repeat expansions, like the CAG repeat expansion that causes HD. And the longer that CAG repeat is, the more unstable that region becomes, causing more complications. And so it's this specific area in yellow that's particularly tricky for molecular machinery to repair. But there are certain tissues and certain organs that have more trouble with this than others. And the blood cells are not one of those cell types. So the CAG repeat you get from your blood test when you get tested for Huntington's disease is very stable. So if they tell you you have either 42 or 19 CAGs when you're 20, you're very, very likely to continue to have 42 or 19 no matter what age you get tested. However, researchers have found that there are certain organs like the liver or the brain where CAG repeats do tend to expand. And so this is a technique used in the lab called a Southern blot. And it's basically a very thin gel where you can run various cells from a person, for example, with Huntington's, in each lane. And so these cells were from a Huntington's patient from various areas of the brain, shown here on the bottom. And you put the samples in the top, and your larger molecules tend to get stuck at the top, and your smaller molecules go further down. And so you can read these blots by looking at the dark spots. And the dark spots represent various CAG repeat lengths. And so what we see is this patient had one copy of Huntington that was about this big, and another larger copy that seems significantly larger, indicating they have Huntington's. But they also have these fainter, darker spots up above that indicate that the CAG repeat expanded in these tissues. And what we can see is there are certain areas of the brain even that tend to show more expansion than other areas of the brain. But what does this mean in the context of HD to have certain areas of the brain or the brain in general have repeat expansions? Well, we know that the CAG repeat length is correlated to age of onset, meaning the more CAGs you have, typically the younger symptomatic onset will begin. However, there is some variability in that. So the patients shown here in these colored in bubbles all have about 42 CAG repeats, but those shown in red develop symptoms about 10 years earlier than expected, and those in green about 10 years later than expected. But why is that? What's causing people to develop symptoms early or later? And one thing that is contributing to that are very small changes in their DNA repair proteins. So the person who develops symptoms later likely has small changes in their DNA repair proteins that causes the genes to expand. And those who develop symptoms later have a significant reduction in that effect. And so if we represent DNA repair proteins with hammers, someone who has lots of CAG repeat expansion likely has a very average hammer. They have a small change that gives them this average hammer, and it doesn't do a very good job of repairing DNA when it needs it. So for example, it might add all of this orange part, and that causes someone to have lots of expansion and get the disease onset a bit earlier. But someone who has very little CAG expansion in their brain might have very small changes in their DNA repair proteins that gives them the best hammer, like for example, Thor's hammer. And this does a fantastic job of repairing DNA when it needs to be repaired. And it might have a much, much smaller CAG repeat expansion. And this causes disease onset significantly later. But DNA repair occurs for everyone throughout their entire lives. So in both of these scenarios, the CAG repeat can slowly get bigger over time. It's just that for some, it will get bigger at a much faster rate, and for others, a much slower rate. So this is what that clickbait headline was referring to. While your CAG repeat size that gets tested by blood in the hospital won't change, there are other cell types in your body, specifically in your brain or your liver, that will have a different CAG repeat length than your blood sample. But what does this mean for HD patients right now? There's currently nothing that we can do about these small changes in DNA repair proteins. So it's not even something that doctors will or can test for, but researchers are currently working on drugs that take advantage of this approach. And they're trying to answer questions to understand, what if they give someone with a less than average hammer a really great hammer? Can they slow how quickly they'll show symptoms? And how long can they delay when symptoms begin to onset? These aren't questions that anyone has answers to right now, but researchers are working on that. Specifically, researchers at a company called Triplet Therapeutics. And someone from this company recently gave a talk at the CHDI HD Therapeutics conference in Palm Springs. And so if you are a scientifically minded individual, you can go to the CHD website and find that talk there. So going back to our headline, hopefully we can understand this a bit better. So the headline indicates that HD causes genetic expansion throughout life. So does the HD expansion keep getting bigger? Yes, but not in all cell types, and certainly not in your blood sample. So hopefully we can understand this headline a bit better. But how can you avoid getting pulled into these clickbait headlines? The answer is that you can get your scientific information from reliable sources. So first and foremost, you can go to your neurologist and verify some of these facts that you might read in the popular press. You can also get your HD news from reputable HD organizations like those shown here, including HD Care symposiums like the one you're attending. You can go to hdbuzz.org. This is a website written by scientists for the HD community, and all of the articles are written by HD scientists that take no money from pharmaceutical companies, so you know they have no interest except getting you the most accurate information possible. And last but certainly not least, if you're a scientifically minded individual, you can go straight to the source. So places like Google Scholar or PubMed are fantastic places. And additionally, oftentimes in these popular press articles, you can look for a link at the very bottom, and they sometimes link back to the original article. So that's another great place to find the original source that this information came from. But overall, if you read something that comes across as really sensational, the first thing you can do is look for another source from this list that either backs up or refutes the claims that you're reading so that you don't fall prey to the clickbait. And so with that, I'd be happy to answer any questions.
So again, just to remind people, you can place your questions in the question and answer bit. We're running a little behind schedule, but I'm curious, Sarah, in order to get science into the news cycle, often the journalists, if not the scientists, want to put a spin on the science. And I'm afraid that without that spin, a lot of the science might not ever reach there. And that's a big dilemma, isn't it? Because in some ways, we would like a more general audience to hear about some of these things. If the work only appears in a scientific journal, how are the public going to hear about promising leads, even if they're not quite as promising as the headline might suggest?
Yeah, so I'm certainly advocating for popular press to get scientific information out to the general masses. But I just think the way that it should be done can be done more carefully, because I think especially some of these headlines are written for people who may have heard of Huntington's, or maybe they knew someone at one point and they'll see that headline and they'll click them. Maybe they won't find the information they're looking for. But for an HD patient to see that, maybe it's like their entire hope resting on that one article. And so I think going to reputable sources, they will give you the information that HD patients and families are looking for, just maybe not in such a sensationalized way.
Yeah. And one of the websites that I think is useful, and maybe you mentioned it, but we could mention it again, is the National Institutes of Health website. And maybe you could say a little bit about that and how people find that.
Sure, yeah. So you can find a lot of scientific information at various NIH websites. So specifically for Huntington's disease, there's information at the National Institute of Neurological Diseases and Stroke, also known as NINDS, because we love acronyms in science. And you can also find a lot of information about clinical trials at clinicaltrials.gov. So if you see something and you think, wow, maybe antihistamines are the next cure for HD, and you go on clinicaltrials.gov and there are any phases of antihistamines for HD research, that's probably a sensationalized trial. So yeah, so government websites, NIH, and including clinicaltrials.gov are great places to go.
Now, if I'm particularly interested in stem cells, which of course I am, but maybe I'm a member of the public, how would I find out about information, for example, to do with stem cells and HD?
I think, Leslie would definitely be a better person to answer this than me, but I think one of the great places to go is to CIRM to see what they're funding. They have a fantastic website where you can see kind of what's coming down the pipeline and what they funded. And you can, Melissa and Mitra can correct me if I'm wrong here, but you might even be able to search for stem cells and Huntington's disease and see what's going on in that arena.
Yes, and thank you for that, Dr. Hernandez. You absolutely can search for that on CIRM's website, and a lot of great information comes up by disease. You can also do that by institution to focus on the rock stars at UC Irvine. Thank you.
And there's a question on the Q&A session from our colleague and emeritus professor, Dr. Golub. Were these studies picked up and marketed by people trying to exploit the promising headlines? And I think what Dr. Golub is getting at is sometimes there are unreputable people that see a scientific headline and start to, develop a fake treatment based on it. And so what are your thoughts about that, Sarah?
Yeah, I think that's terrifying. So I hope not. I certainly hope not. I haven't heard anything related to these two trials, but related to that, I would also caution a lot of these articles are also either funded by or contributed to by pharmaceutical companies that have a stake in some of these things. So you want to be careful with that, right? Like the pharmaceutical companies will have an interest in trying to make their drug sound like it's totally going to work, when in actuality, maybe the clinical trial came very significantly close, but it didn't cut the mustard. So those are other important things to make sure that you get your news from reputable sources that don't have a financial interest in making it sound like we cured HD.
Sarah, thank you very much. I'm sorry we don't have more time for questions, but thank you for bringing to our attention a very important issue in the field of clinical trials and research development. Very important. Thanks a lot.
Thank you.
And now we've flown him all the way from Massachusetts. It's a pleasure to welcome Paul Dennis, who's a Master of Fine Arts, which I'm told by my colleagues here is like a doctorate in the arts. And he trained at the Juilliard School, which is a name that's familiar to most of us. And I think his talk is going to be really interesting because his research uses patterns of movement to develop intervention programs for those living with HD. And I think this is a really interesting area, and this should be an exciting presentation. So Paul, thank you so much for joining us today.
It's wonderful to be here. Thank you very much, Peter. And thank you, Melody, for extending the invitation. I really appreciate it. And I hope that what I present here is encouraging and fun and causes some movement. So having said so, I'd actually like to start off with a little bit of movement. So I know there are many of us seated, most likely seated in chairs, and or lounging or sitting back on a couch or wherever you are. And it's hard to talk about movement and dance without actually doing a little bit of it. You don't have to show your video of dancing or anything, but I'm going to encourage you to do a little bit of movement with me as we get started. For the work that I do, and I will continue to give you an idea of what that is. For the work that I do, I work on trying to have total body connectivity through a very specific system. And one of the things that I do with those with Huntington's disease, who I'm teaching a class for dance for Huntington, is something that I'm going to invite you to participate in. I call it lemon and starfish. And that is, imagine you're a starfish and you're just going to spread out as you can. If you're able to spread your arms out in front or to the side and your legs, and then bring them all into a small, as if you're sucking a lemon really, really tight. And then just move a little bit. We've been sitting down in chairs. I am so zoomed out these days, sort of spread like a starfish. Please join me if you can. Do your best, or come right back into a small lemon. And one more. Go on out, nice and large, and come back into a small lemon, really, really tight. And one of the things that does is you get a sense of two differences. You get a sense of tension and binding and strength on one, and then lengthening and rising and lightness on the other. And if you find yourself like me in front of a camera all day, Zoom teaching or Zoom learning, just getting up and allowing your body to go through those two extremes, those two tensions, is really rejuvenative. So I'm going to share my screen and go on with what my research is. So here we go. Dance for Huntington's disease. And the subtitle here for this slide is Neuroplasticity and Dance. Dance is a complex sensory motor activity. It involves the integration of rhythm, of spatial patterns, to synchronizing to external stimuli, and whole body coordination. One functional neuroimaging study measuring brain activity on live dancing using PET imaging found that there is activity underlying the awareness of body position in space. In other words, how and where our bodies are in relation to itself and in relation to space. And also an awareness of underlying somatosensory processing, sort of common to different dance conditions. So whether you have a certain rhythm, whether you have to navigate high, low, a certain space turning around, there was a lot of measurable activity in parts of the brain. And these studies suggest that regular activities such as walking or reaching, stooping, etc., scale up, they ramp up to rhythmically timed, spatial, patterned whole body movement. Now, while regular exercise is frequently recommended for Huntington's disease and other neurodegenerative disorders, there have been no studies examining the use of dance to positively affect the quality of life for those living with Huntington's disease. So that's what we've been doing. We've been studying how would dance affect the quality of life for those living with HD. The work of Rudolf Laban and Irmgard Bartenieff, which is called the Laban-Bartenieff movement system, is one of the most comprehensive systems of human movement analysis today. Rudolf Laban was a dance theorist and a teacher who studied human motion, and it provided sort of the intellectual, the scientific foundations for the development of Central European modern dance. And his analysis of forms in movement is known as choroitics. It is a non-personal scientific system and practical study of various forms of harmonized movement. So you might ask, well, what is harmonized movement? A slide later on will show that, but imagine the same way as when you listen to harmony in music, a certain scale, certain tones, how pleasant it is, how you could almost tell where it's going, where it's come from. Well, movement also has a certain harmony to it that's applicable to all human motion. So choroitics had been used in clinics and psychiatric settings with clients having numerous issues, including autism, on the autism spectrum disorder, Down syndrome, traumatic brain injury, and learning disabilities, etc. Laban movement analysis is specific and adept in describing what moves, where it moves, how it moves, the why of movement, and the relationship of the mover to him or herself and others and the environment. So one of the aspects is the body. So the body, there are these four broad terms that are, or characters that are used to describe any type of movement. The first is the body. It describes what's moving, what body part, the coordination of that body part, the basic action of that part or the whole body, walking, jumping, sliding. The next is space. Where is the body moving in space? What direction is the movement? Up, down, forward, backward, sideways, open, or across? What are the planes of movement that occur, whether the vertical or sagittal, horizontal plane, as well as where the movement is in relation to the center of the body? Is it far reaching and distal, or is it close to the core? Or is it in somewhere in between? Another category is shape. It describes the shape of the body itself moving in relationship to its surroundings and with others. Is it rising? Is it expanding? Sometimes it reflects the why of the movement. It's shrinking, condensing. We often observe shape when we notice whether it's the body is spreading, or like I said, rising and sinking. So that's one of the ways, one of the categories. And then there is effort. And effort describes how the body moves. So it's the qualitative aspect of movement, such as is the movement light or strong? Is it sudden or is it free? Effort sort of reflects the inner attitude of the mover towards the movement. The categories of effort are weight and space, time, and flow. Irmgard Bartenieff, she also was a dance theorist, a dancer and choreographer, and a physical therapist. And she was a leading pioneer in dance therapy. She began studying with Laban after receiving a degree in physical therapy from NYU back in the early 1940s. And she went on to work with polio victims and as a dance therapist and research assistant in nonverbal behavior. She developed a system of observing and notating behavior using Laban movement analysis techniques. And then she developed her theories in what is now called Bartenia fundamentals. And that work describes things like dynamic alignment, breath support, core support, rotary factor in gestures, initiation, where does movement come from and how does it sequence, the spatial intent of movement, the center of weight and transferring of this weight, the effort intent, the amount of energy and dynamics behind a certain movement, and developmental patterns in support of level change. So you may realize with all of this that this Laban movement system is a method for generating, describing, documenting, and analyzing all varieties of human movement. This is the system that I present in my dance curriculum. It offers those who participate a range of dynamic possibilities for movement, for repatterning and improving functional mobility and cognition. And it's what we call dance for Huntington's disease. Earlier in a slide earlier, I talked about neuroplasticity. I mentioned it. Well, neuroplasticity is the capacity of the neurons in our brains and neural networks to change their connections and their behavior in response to new information, to sensory stimulation, to development, to damage or dysfunction. And the features, neuroplastic features, are specific for the therapeutic modality. For example, listening to music improves global neuroconnectivity, while playing a musical instrument, singing, or dancing increases gray and white matter in the brain and alters neurochemical levels, including endorphins and dopamine. We heard about dopamine earlier in one of the presentations. So consequently, music and dance movement therapies in conjunction with conventional clinical interventions have the potential to affect cognition, psychiatric and motor functions for persons with progressive neurodegenerative disease. There have been studies about with patients who have multiple sclerosis and Tai Chi, or dance and Parkinson's disease. But once again, there have been no studies yet on dance and Huntington's disease. Laban's work envisioned a dynamic space. He identified relationships between single spatial appearances, causing movement to follow definitive paths. And he found that there was a unity of movement and space that can be demonstrated to show a natural order of sequences. Remember, I talked about space harmony or movement harmony earlier? That's really what he discovered, that there is a natural order of sequence and laws governing the order of movement sequences. And these laws give us orientation in space. And as we move, there are actual trace forms. If you could imagine, there are ways in which movement can be traced through space that, that follow a natural order. And this movement is, in a sense, a living architecture made up of pathways, tracing shapes in space. So if you could imagine the octahedron or icosahedron that this person is inside, imagine dancing through, tracing these platonic solids as a way of harmonizing movement. There are some stable dimensions, height and breadth and depth. And even though they're challenging in the life of Huntington's disease patients, the goal is that once we, with mindfulness, access these high and deep lines of spatial intent, that our mind, so this is the neuroplastic part of it, oftentimes, or most predominantly, we think of the mind affecting the body. Neuroplasticity sees it as a two-way street, that the body can actually affect the mind. I'm sure you've seen there are various YouTube sites where you see someone who has some sort of movement disorder, just walking down a corridor, say in their home, and then putting music on and walking down the corridor, and how the symptoms of that disorder changes dramatically with rhythmic and spatial patterning. The same thing is what has been discovered with MS and Tai Chi. With rhythmic spatial awareness, the symptoms can actually, I wouldn't say be cured because there is no cure, but the idea is the neuroplasticity properties of the brain can actually circumnavigate. The aim here is to lessen the symptoms and increase the functions of the body by the body telling the brain how to fire, how the synapses and the neurons connect. There is a pretty famous actor who has Parkinson's disease and is a very good ice skater, ice hockey player. Once that person gets on their ice hockey skates, you will have no idea that they're suffering from the symptoms of Parkinson's disease. And that's what Norman Doidge in his book, How the Brain Heals Itself. That's how he describes the possibility of the body to teach the brain how to fire, so that we can circumnavigate some of the dysfunction to actually find functional capacity in our movement. So I'd like to do a little bit of movement again, because I'm talking here about this dynamic space, and to give you an idea of what that might be. So I'm going to stop sharing just for a second and go back to, oh, here's my video. Again, if you're sitting, I'm going to invite you to do a trace form within an octahedron. And what that will do is we're going to go high and deep, advancing forward and back, retreating, crossing, enclosing, and widening, opening. You can use any hand that you'd like, both if you'd like, or if you would like to just watch as I do it, that's just fine. Because watching actually fires neurons in your brain almost as if you're doing it. So I'm actually going to put a little bit of music on. Hopefully you can hear it. It won't be too loud, but for the most part, just to enjoy the rhythmic part of the movement. So here we go. So we're just going to go high. We're just going to stretch up 1 or 2 arms high. And then deep. And then cross or enclose. And widen or open. And if you can change arms, do the same thing. High and deep. Cross and open. So one more time and we'll add another element, high, deep, advance forward, Other arm. And deep. And close. And widen. Ah, should have done advance. Advance, forward. And back. So that might seem really simple. High, low, cross, and open. But that is part of what in LMS called the defense scale. And it's this most stable part of moving through a scale. There is up and down, forward and back, and close and open. And that's within this certain framework. So in closing, I'll just go back to the slide and close off here. In closing, dynamic space, rhythm, spatial awareness, the inter and intrapersonal experience of dance, the patterns and imagery, the tensions and release is fertile ground in which movement flourishes. Movement is the life of space, and all movement is this change. There's been a great expansion of arts and medicine programs across the nation in recent years, building on a greater willingness to explore the efficacy of alternate treatment modalities. I'm hoping that the work that we do with dance can build momentum for alternate treatment for Huntington's disease, those living with Huntington's disease. Thank you. I'm eager to answer any questions.
Paul, thank you so much for that wonderful presentation. I forgot to warm up before this symposium, so I didn't stretch. But there's actually a question that we can maybe answer from Hertwig Lange. And he points out that there are a number of scientific studies looking at video game-based training and other aspects in terms of HD. And I wondered if you've seen those studies and how they relate to your own work. And maybe also you could go on and tell us, how did you become aware of HD and how did you start working with people who have HD?
Thank you. Great question. Yes, I am aware of that work with video games, and I think it's phenomenal work. I think it's one of the first studies that had been coming out, and it showed great promise for how movement, in fact, can affect neurodegenerative disease. And since then, there have been other studies that have started to do the same, not quite as specific as what I'm aiming for here. So this will answer another part of the question. Part of the movement that I will introduce to a class is not just memorizing the movement or the joy of the movement, but also the awareness of it. So for example, when I cross my arm, when I take my arm across my body, that feels different. There is a very different sensory motor connection happening between crossing and enclosing and widening and expanding. Instead of just doing that as an exercise, dancers and Dance for Huntington spend time asking and being aware of the feeling of this crossing. So
and is the notion that in doing that, you're stimulating different parts of the brain and therefore helping them stay healthy?
Yes, it's an awareness. So it's not just the movement and not just the rhythm, but the awareness of it, which is why Tai Chi has been shown to work really well with patients with MS. It's the awareness of the movement, of this transferring of the weight. So I got involved as I was studying for my certification for Laban movement analysis. And at the end of the study, you have to do a thesis. No one, you know, it's almost like a doctorate, like you were saying, Peter.
Yeah.
So I had this huge thesis to do. And I was just about looking for something that would grab my attention. And I had a few things. Then I received an email from Dr. Garg and Dr. Demoto at Tewksbury Hospital in Massachusetts, who were the doctors in charge of the different therapeutic modalities for patients with brain injuries. And I answered, you know, here's the work that I'm doing. I believe that it could work with traumatic brain injuries. And I was invited onto the campus. When I was there, I saw people who had different types of brain injuries, from stroke to vehicular accidents and whatnot. But it wasn't until the last floor that we were on that someone who is living with Huntington's disease came bounding down the aisle and said, "Hi." And I was introduced as Professor Paul Dennis, who was a dancer. And the person says, "I love to dance." And he just went bounding away. And I thought, oh my gosh, his movement, the choreography of it. And it was, you know, someone with Huntington's. And I said, that's the population I want to work with. The movement really appealed to me, really got me thinking about how my movement, and as I learned about movement analysis, can help those who have challenge with movement disorders.
So Hertwig, I've posted some more of his comments and answered them live. And one of the points he made, and I can't quite catch where it is now, is that dance is really important for them and helps with rehab, and they really appreciate it. And I guess that one of the things, Paul, you're obviously someone who's very trained in this area. Do you know of other programs around the country that people might reach out to where they could get involved in this? Or is it simply, you know, go to a dance class and get involved? What would your recommendations be there?
I've been working on Dance for HD since maybe for the past 9 years. And we've just gotten funded to actually have a clinical trial so that,
fantastic.
well, unfortunately, with COVID-19, it is impossible to have a class with, you know, face-to-face. So we have postponed the beginning of the trial until next year, late next year, and see how that goes. So there is no official Dance for Huntington's disease that's there as yet. And hopefully when we finish the clinical trial, we have some data to validate what dance does. However, having said that, any dance class. So if someone is interested in movement, again, I would recommend movement with awareness, like Tai Chi, because there is a certain, again, awareness of where your body is in space, and not just physical therapy. And I'm not saying that physical therapy is, but it's just different. So any dance class, and if you can go with do partner dancing, even that's even better, because now you're not only being aware of your body in space, but you're being aware of someone else, and it's another level of awareness that is absolutely amazing. So yeah, go take any dance class.
Now, I wondered if you would mind doing this for us. It's been my experience interacting with the HD group here in Orange County that the families are always interested in their loved ones getting involved in clinical trials of whatever nature. Although obviously I don't know all the details of your clinical trial, is there a way in the age of Zoom that people here could register for it, maybe film doing some stuff that could be sent to you, or what's your sense of that?
Yeah, I'm actually, thanks for asking that. I'm actually considering it because we don't know what tomorrow will bring regarding face-to-face activity or being in a studio as far as COVID-19.
Yeah.
Part of the clinical trial involves 4 tests, and that will be the most challenging part to navigate because those who are participating will go through a number of tests, first pre-testing. 12 weeks later, we do a second set of tests. And that 12 weeks later is the beginning of the intervention period where we have 12 weeks of dance. At the end of the intervention period, we have a third test. And then 12 weeks later is another test. So we'll be measuring the differences between the tests. That's the most challenging part if we were to do it remote. If I can find an answer to that, that'd be great.
I tell you what, in the Stem Cell Research Center here, we have a number of engineers that use robotics to test people's movements to help them. And one of our colleagues is actually in the School of Dance in the department, or the School of Arts in the Department of Dance. And maybe I should put you guys in touch. But there's another question on the on the thing about, can you transfer the training to web-based? And I have a suggestion for you. Would you be willing to tape a little session and give it to UCHD Care and see how people like it?
Sure, why not?
Yeah.
I've done a couple of sessions. I don't know if they were recorded. They might have been. But the truth is, yes, I can. Once I develop the whole protocol, I'm sure that whether or not someone is part of the trial, I am positive that they will benefit from it.
Yeah.
So developing the protocol, putting it on something like the Vimeo, something that's pretty stable, if not YouTube, and having it available to participants with Huntington's, families with Huntington, it'll be my pleasure.
You could give us a little sample.
Pardon me?
You could give us a little sample.
I sure can. Wait, you mean now?
No.
Oh.
On the video. Hey, Paul,
Yes.
we're a little bit short on time. We're running late. Thank you ever so much. This was really interesting. And I think it just points out the importance of music and movement in mental health, really, and why it could be so important for patients with HD. Thank you ever so much for joining us.
It was my pleasure to be here. Thank you for having invited me.
So now I'm going to hand over to my colleague, Leslie Thompson, who's going to introduce the next guest.
Hi, all, and thank you, everyone, and thank you, Paul. That was amazing. I really enjoyed that. Yeah, so it's a real pleasure now to introduce our next speaker and keynote today. Again, this COVID presented an opportunity to have one of our field's rock stars to come here and give a talk. And so Blair Levitt is a neurologist and professor of medical genetics at University of British Columbia. He's been a long-standing researcher and clinician in the field of Huntington's disease. He was the former co-chair of the Huntington Study Group. He's the director of research at the UBC Center for Huntington's Disease. And he's been involved in just countless number of clinical trials. He runs the HD Tissue and Brain Bank at UBC. He's been involved in Track HD. He's one of the co-PIs on Enroll HD, HD Clarity, and he, along with Sarah Tabrizi, co-led the first Ionis trial with ASO treatment with Huntington lowering, and is now involved in the new Roche trial, which you'll hear about from him. He got his medical degree from McGill University, internship at Columbia, did neurology residency and fellowship at Cornell and Harvard, and then neurogenetics fellowship at UBC, and had started out working with Michael Hayden, who many of you know, and then has been independent since then, doing both basic science and really a lot about biomarker discovery for Huntington's disease, as well as clinical trial work. And I have the privilege of being co-editor-in-chief of the Journal of Huntington's Disease with Blair, which has really been a pleasure. And so it's an honor to introduce Blair Levitt. Thanks, Blair, for joining us.
You saved the biggest honor for the last one, which is the fact that you and I have been able to work together for the last, is it 8 or 9 years? I don't know, it's flown by.
Thank you.
I'd just like to thank you so much for giving me an opportunity to chat and to talk about some of the clinical trials that are ongoing in Huntington's disease. Hopefully, we had a little bit of a technical problem and I've had to switch computers. Hopefully this is working. We haven't had a chance to run through. Can you see my slides?
Yep, working fine.
All right, great. So I've been tasked with giving an overview of clinical trials in Huntington's disease, and that's actually a big task right now because it's a very exciting time. There's a lot of activity going on, and I think there's a fair bit of excitement about the types of trials that we're moving into. We've really moved beyond sort of the initial phase. The gene was discovered. We learned a lot about Huntington's disease, but most of the therapies that have been tried in the last 25 years have been things that were on the shelf already, previously developed drugs or therapies or supplements. But we're really now moving into a time where we're looking at directed therapies and really, most excitingly, some of the genetic therapies. So when I talk today, I'm really going to be focused primarily on the current genetic therapies that are in clinical trials. I'm going to also have a brief discussion of some of the other trials. Sarah did a great job of introducing some of this in her talk. And then just at the end, a brief overview of some of the future approaches that are close to clinical trials, but are still in the preclinical phase that I'm quite excited about. And certainly, I don't have to tell this audience that stem cell therapies have great promise and that CIRM is a very exciting opportunity. And certainly Leslie has been leading the field there. So I think you're well aware of that. And then hopefully we'll have significant time at the end for questions and answers. And I hope to be able to answer any of your questions. So these are the studies, the current HD genetic therapy studies that I'll be talking about. Some of them led by Aunis and Roche. Wave Takeda has 2 trials ongoing that I'll talk about. And then we have our first true gene therapy trial starting in humans led by Unicure. So before I start, I just wanted to make sure we're sort of all on the same page. And this is sort of, this is what we call the central dogma of molecular biology. And it's really how genetic information flows. How do our genes create the components of our body? And when we talk about genes, we're really talking about DNA. And it's the code of the DNA that gets transcribed into what's called RNA. And RNA is the messenger between the genes and the proteins. And the proteins are the building blocks, or some of the building blocks of our cells. And so it's really important that we understand this pathway and understand this so that we can understand the current genetic therapies that are in development. And specifically when we're talking about Huntington's disease, we need to understand that we know the cause. The cause is an expansion of CAG repeats within the Huntington gene that produces a protein that we call mutant Huntington that has an expansion of a specific amino acid, glutamine, so a polyglutamine stretch within mutant Huntington. And it's believed that it's likely that abnormal protein that causes brain cells to become injured. So we start with the gene, an RNA is made, the RNA makes the protein, and the protein is probably the bad player that causes the disease. And there's a large number of different ways that this abnormal protein can injure cells, brain cells and other cells. And the field has been very active in studying these, what we would call downstream pathways. But what I'm going to talk to you about today is really focusing on that main player, that bad character, the mutant Huntington protein. And we abbreviate the protein Huntington with HTT. So if you see mutant HTT or mHTT in any of the slides, what we're talking about is the abnormal protein that is produced from the gene, from the expanded CAG repeat. And there's a really basic and pretty simple hypothesis behind Huntington lowering therapies. And that is that if we can prevent the generation of this toxic protein, that should prevent all of those downstream pathologies, all of those downstream toxic effects. And hopefully, if we can give it early enough, delay or prevent the onset or slow the progression of the disease. So it's a theory, it's a hypothesis. It's one based in a lot of basic science and our understanding of how disease occurs. And one of the things I'm going to talk to you about is antisense technology. And antisense technology actually makes sense if you understand what the words mean. And when we say antisense oligonucleotide, or ASO, what we're talking about is an oligonucleotide, which is basically artificial DNA. It's a stretch of nucleotides. Oligonucleotide is just a stretch of nucleotides. So it's a little stretch of artificial DNA. And it's antisense, meaning it's the opposite of something in the body. Let's say a piece of RNA. And when it's opposite, or antisense, it will bind to that in a very specific way, in such a specific way that it won't bind to any other nucleic acids in the body. So that's what we're going to be talking about, ASOs, and that's where the name comes from. That's what it means. And this is one of the forms of what sometimes is called gene silencing approaches. And we're going to talk about ASOs. We're going to talk about some of the other approaches a little bit later. But really, we shouldn't be calling it gene silencing. Specifically in the case of Huntington's disease, we should be talking about Huntington lowering therapies. We don't silence genes. We try and decrease their expression. And there's a number of ways we can do that. There are what are called small interfering RNAs. They target the messenger RNA, so just before the protein is produced. Antisense oligonucleotides target a longer piece of the RNA called pre-mRNA. And then there are other approaches that I'll talk about at the end that actually now are targeting the DNA itself. And these are zinc finger proteins and different kinds of genome editing, CRISPR-Cas9 approaches. So we have small interfering RNAs, we have ASOs, and we have genome editing, all of which are potential ways to lower Huntington. In addition, we can also, and I'm not going to really talk about it, try and decrease the amount of protein by increasing its clearance in the cell. And that's quite an exciting approach, but none of those therapies are yet in clinical trials, so I'm not going to really talk about that today. So how does an antisense oligonucleotide, or ASO, work? As I said, we've talked about the basic pathway of genomic information from DNA to RNA to create a protein. When we make an antisense oligonucleotide, we make it to a specific RNA. It will then bind. It now forms an RNA-DNA hybrid, a piece of nucleic acid that shouldn't be there. And the cell actually recognizes that. There's an enzyme called RNase H that says, this is wrong, there shouldn't be RNA and DNA stuck together, and it chops it up and prevents it from producing the protein. And that's how an ASO decreases the amount of protein. It interferes with the messenger, the RNA, between the DNA, the gene, and the protein. And when we're talking about ASO therapy for Huntington's disease, now we have the Huntington gene that makes Huntington RNA, and we have an ASO, which is again a stretch of artificial DNA that's antisense. It's opposite to that RNA, and it'll only bind to that Huntington RNA, none of the other RNAs in the body, none of the thousands and thousands and tens of thousands of RNAs in the body. By doing that, it will prevent the production of the protein. ASOs have been around for a long time, actually, and there's a number of advantages. They're very much like traditional therapeutics. They're what we call diffusible, dose-dependent, stable, and reversible. In other words, if you can get them to a cell, they get taken up freely. The more the cell takes up, the more it knocks down the protein. They are relatively long-lasting. They have a stable effect over months in many cases. But importantly, they do get broken down by the cell and they disappear over time. So they are reversible. And that's a really important safety factor. We can give these, they can lower the protein. We give more, we get more lowering. But if there's a problem, we can stop giving the ASOs and it's reversible. So that's a really important factor to understand. How do we get these kinds of drugs into the central nervous system, into the brain? And what we really care about is lowering Huntington in the brain. We do this, and I think Melody gave a bit of an insight into this. We do what's basically a reverse spinal tap. We put a small needle between the vertebrae into the space where what we call the CSF, or cerebral spinal fluid, is. We take off some CSF and then we replace it with artificial CSF containing the drug. And remarkably, even if we put it down in the lower back, it gets all the way in equilibrates throughout the CSF all around the brain. And actually, the CSF penetrates deep into the brain tissue. So even though we're administering it in the lower back, within 30 minutes, it will equilibrate through the entire CSF volume and get to anywhere in the brain the CSF goes. And this is how this agent can get into brain cells. So before we talk about trials, there was one other really important identification. As I said, what we really want to do is lower Huntington in the brain. That's what we want. But we can't measure that in a clinical trial. We don't have any way yet to measure brain levels of Huntington. There are some really exciting approaches using PET, and CHTI is developing some really exciting PET ligands, but they're not quite ready yet, and we don't know that they're going to be useful, but it's a great approach. The best we have is something indirect. We can't look at the brain itself, but we can look at the cerebral spinal fluid that the brain floats in, and we can measure the amount of abnormal protein in the CSF. And there were some really important papers that came out just at the start of the ASO trials from a couple of groups. And this is just some of that data. And you know, I'm a scientist, so I got to show you some hardcore data. This is patients from London and patients from our group in Vancouver. And this is patients that are affected by Huntington's disease, and showing that you can measure this abnormal protein, mutant Huntington, in human CSF. And you can also measure it in pre-manifest, although you can see it's generally lower. In controls, people that don't have an expanded gene, there's no measurable Huntington. So this proved that we can now measure mutant Huntington in the CSF, and that might be a surrogate marker. But how do we know? Do we know that the levels in the CSF are equivalent to brain levels? We don't know. But the best data we have came from some really cool animal studies. And one of those studies, we took an ASO and we put it into a mouse brain, and we knocked down the amount of Huntington in the brain. And we saw a really nice, what we call a correlation. As the amount of Huntington in the brain went down, the amount of our measurement in the CSF went down. And there's a nice correlation. So yes, there seemed to be a correlation, at least in rodents, with the amount of Huntington in the brain and the amount of Huntington in the CSF. So at least this is the best we have, but it is an important marker, and it actually came just in time for the first trial. And that's what I'm going to tell you about. This was originally run by Ionis, and the drug was originally called Ionis HTTRX. Subsequently, Roche called it RG6042, and now it's called tominersen. And I'm going to tell you, this has now been published, and this is the first Huntington lowering trial. And we would say it's a successful trial because it was a phase 1/2A trial, which is really a safety trial, and it was shown to be safe. So that was success. We showed it was safe. Just a quick slide. I don't expect anyone to read any of this, but I want people to really understand that this trial is the result of many, many years of extensive development, both by the company, but also by a large number of academic collaborators all around the world. And it was an incredible group, CHDI, Ionis, Roche, and scientists from around the world working to get this first trial underway. And it's been a really amazing collaborative effort to date. So again, 10 years of preclinical work, first patient in September of 2015. So this initial trial was done at 9 sites in the UK, Germany, and Canada, and was run, I wasn't co-PI, Sarah was the overall, Sarah Tabrizi in University College London was the PI. And thank you, Leslie, for making me co-PI, but I was just one of the investigators. Sarah was our fearless leader. And again, it was an intrathecal administration of an ASO, is what we call a bolus push. So basically we take off CSF, and over 90 seconds, we push in 20 cc's of the drug. We looked at a number of outputs, including mutant Huntington in the CSF, but this was a first-in-man trial, a phase 1/2A trial that we call. And really the outcome measure that was most important was, is it safe and well tolerated? And not to ruin it, but yes, it was safe and well tolerated, so it was a successful trial. So tominersen is a ASO that targets human Huntington. It is not specific to one or another. It targets all Huntington. So we don't want to lower it too much. We targeted about a 50% lowering in CSF. Again, it's given by an intrathecal slow bolus push. And based on our preclinical studies, that 10 years of work, we knew that it had a fairly long action. but we didn't yet know, did it need to be given monthly, every 2 months, or even longer? So first trial was started. This is actually a very happy neurologist after the very first patient was enrolled and the infusion was just done. And this started in September of 2015. We started out with very low doses of the drug. And this was a safety trial, so we wanted to be sure before we moved on to higher doses that it was safe. So we repeatedly dosed a very low dose, and the investigators were blinded. We didn't know if they were getting drug or placebo, but an unblinded safety review was done by what's called a Data Safety Monitoring Board, or DSMB. And before we could go to the next dose, they had to see that the cumulative dose was safe, and it was. So we moved to higher dose, and eventually we got up to what we felt were going to be effective therapies, 60, 90, and 120 milligrams. And this was given monthly. And to cut to the chase, it was safe and well tolerated, but really much more exciting, we did see a dose-dependent decline in CSF mutant Huntington levels. And you can see at the two highest doses, a range of around 40 to 60% decline. And importantly, this was the level of lowering of mutant Huntington in all of those preclinical animal studies that correlated with an improvement in the Huntington's disease phenotypes of mice and other studies. So this is what it looked like longitudinally over time. And you can see that we didn't really reach, this was the last dose at 85 days. It was still going down. So we weren't quite sure what would happen if we continued those monthly doses. But it looked very promising. And again, the summary of the trial results were this: it was safe and well tolerated. There were no serious adverse events. Nobody dropped out. All 46 subjects continued on, and all of them eventually got placed in open label, which means they were all getting active drug, and they've now been followed for over 2 years on active doses of the drug. And again, quite an exciting secondary finding was that there was dose-dependent lowering of mutant Huntington in the CSF. So this has now been published in the New England Journal. And the bravery of these first 46 volunteers can't be overstated. This was a first-in-man trial. These individuals from these various countries participated not knowing that it was safe, not knowing that it would be well tolerated. There was a lot of concern about repeated lumbar punctures. But based on this, now there is a very large and robust program now being led by Roche to look at a number of studies. All 46 of those volunteers went into what's called open label, where they were getting active drug either monthly or every other month, and they were followed for many years. There was a natural history study that matched those individuals and followed non-treated people, 2-to-1 matching over a similar period of time. That's now complete. And those individuals in the natural history study are now in an open label study. So open label studies are really important. They allow us to follow people on active drug for a long period of time to look for safety measures. So there's been continuous safety monitoring of this administration now for many, 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 many months. Really exciting, Generation HD1 is the phase 3 efficacy trial. So the first trial was really safety and tolerability with some exploratory measurements to look at whether it was affecting CSF mutant Huntington, whether there was any changes in clinical scales. But really, all it did was provide the groundwork for a proper efficacy study. And that's what Generation HD1 is. It's a phase 3 trial designed to see whether this treatment prevents or delays progression of Huntington's disease. It's a 25 months follow-up of now almost 800 individuals on 2 different doses compared to placebo. So that's now complete. The recruiting is complete. Again, there's almost 800 individuals worldwide, and we'll know the results of that in roughly 2 years' time. So very exciting. There are other trials that are also using similar approaches. Another very exciting approach is Wave Life Sciences approach. They're also using an antisense oligonucleotide. In this case, they have a slightly different way of doing their chemistry. They call it stereopure nucleic acids. And this is in early clinical development. So they're currently sort of in that phase 1/2A trial stage. And they're specifically targeting a type, a genotype, so a specific sequence in the genome that some HD patients carry and some don't. So you have to have just the right genotype for these drugs to work. And based on preliminary work, it looked like that if you had 2 of these ASOs and you screened a population, you might be able to treat about 2/3 of the population. Might end up being a little bit less. About half the people will have just the right type of genotype for these drugs to work. Unfortunately, it won't work for everyone. And that's one of the drawbacks of this approach. But the advantage is, if it works, it will only be targeting the abnormal allele and shouldn't affect the normal Huntington allele. So this has now been underway since, I believe it started in July of 2017, originally 48 participants. Again, it was a increasing dose of this drug. There haven't been any published results, but there has been a press release from Wave in which they analyzed the initial study, at least with one of their 2 ASOs, and they did find a reduction in CSF mutant huntingtin, which is great. My understanding is in their study, it was also safe and well tolerated, which is great. But unfortunately, the effect size was very small, and the reduction was somewhere in the range of 12 to 13%. So not a huge reduction, but they also had relatively low doses. So based on this, the press release reported that they'll be trying higher doses, and I believe that's underway, and we would look forward to some additional results from Wave in the near future. Important to say, again, one of the endpoints that they would be looking at is safety and tolerability, but also to look at the level of mutant huntingtin being knocked down. So we've talked about ASO approaches, but there's another approach that's now in clinical trials, and that's targeting the Huntington mRNA using a small interfering RNA, in this case what's called a microRNA. And this is an approach by a company called Unicure, and it uses a different way of getting the gene therapy into neurons. In this case, it's being directed directly into the brain, and it's being carried by a virus. AAV stands for adeno-associated virus, and these are recombinant viruses, so they're not naturally occurring. They're essentially a virus where we've taken out everything that makes it pathogenic, makes it a virus that causes disease, and we can put in our own cargo. And so this allows us to put in this microRNA, and the microRNA targets the Huntington messenger RNA, much like the ASO does. But in this case, we're putting in the gene that produces it, and that gene can keep producing it. So a single injection into the brain will produce this microRNA for a very long period, potentially for life. So it's a one-shot deal. That's a great advantage in that it doesn't need to be repeatedly administered. It has some disadvantages, and it can't be reversed. So Unicure has a development candidate. It's an adeno-associated virus vector that's putting in a microRNA called AMT-130, and they use a particular type of vector called adeno-associated virus 5 or AAV5. There are a couple of other AAV approaches that are very similar, being developed by Spark and Voyager, but really Unicure is the first one. And again, a little different from the ASO. It's being injected directly into the brain, so it's a neurosurgical procedure and multiple injections. The advantage is it targets deep structures in the brain very well, but may not cover the whole brain. ASOs have very good coverage in the cortex and much of the brain, but maybe not quite as much in the deep brain structure. So they both have advantages and disadvantages, and time will tell which is more effective. So again, this is a first-in-man trial. The most important thing is it safe and is it tolerated? But they will be looking at a number of outcome measures. This will be a randomized, double-blinded study in which patients and their caregivers will not know whether they're getting active drug or a sham or imitation surgery. So this will be 2 different doses of about 26 patients. It's now started. These individuals will be followed up for 18 months, and then my understanding for another a total of 5 years. But after 18 months, those individuals who got the imitation surgery, I believe, will be given the option of active virus. So those are the current Huntington lowering therapies that are in current clinical trial. There's good evidence for safety in the ones that have gone far. This first Ionis HTTRX trial is now complete and published. And I think these approaches do hold great promise for the future treatment of Huntington's disease. It is, but in no means should we stop. We need to continue to develop effective therapies, and it's very likely that even if we have something that has an effect on progression, we'll need likely combinations of therapies in the future. So what are some of the other things that are undergoing? I mentioned a lot of excitement about targeting even more upstream, targeting the DNA itself. There's an exciting approach using what are called zinc finger approaches. And zinc finger approaches are molecules that bind directly to DNA and prevent the mRNA from being produced. And Takeda, it has one in development for Huntington's disease, and there's a fair bit of interest in that. It's exciting because that Takeda zinc finger approach is also specific. It binds to the expanded part of the DNA and should be only targeting mutant Huntington. So that's quite interesting. You may have heard about genome editing, CRISPR-Cas9. It's sort of the molecule of the decade, and it allows us to actually change the DNA in cells. And there are a number of different approaches. I've started a company called Incisive Genetics, and we are using Huntington's disease as our initial target for a lipid nanoparticle way. It's a different nanomedicine approach to getting gene editing into cells. So this is not quite ready for HD clinical trials, but very exciting in the future. And of course, there are other approaches. Sarah did a great job of introducing triplet therapeutics. They're targeting instability, those changes in the DNA, and they have a natural history study called SHIELD, which hopefully down the road will lead to a therapeutic trial. Stem cell transplantation and regenerative medicine is an area that desperately needs to continue to be developed. We need more and more basic science to help translate that, the real promise into clinical trials. And of course, we do need to get those clinical trials up and running. Signal and Nexon, Signal, a trial by Vaccinics that recently was completed, and an Nexon, a trial that's currently just started, both targeting inflammation in the brain. Unfortunately, the Signal trial from Vaccinics just released its primary outcome measures, and it was did not have a significant improvement on those primary outcome measures. But we're still evaluating that trial, and we'll see whether or not future trials are warranted. And then Nexon is an early stage clinical trial that we don't have any data of yet. So that was a whirlwind view of what's going on in trials. I wanted to leave as much time for questions and answers, so I'll open it up for questions and thank you for your time, and thank you, Leslie, and the group for this opportunity to speak.
Thank you so much, Blair, for coming and joining us. We really, really appreciate it. I'll hand it over to Peter, but thanks also for mentioning the SERM trial, work and stem cells. And all of this work that you guys have done so incredibly beautifully with all these trials, and the information we're learning is going to apply so importantly to everything everyone, the whole field is doing to advance therapies for Huntington's disease. So, and thank you for being an advisor on our SERM work. And
I'm super excited about it as well, Leslie, as I said.
And the Huntington lowering is just incredible how quickly this, it's a lot of years, but it's relatively gone very quickly, and it's just beautiful. Peter, do you want to take over from here?
Blair, thank you for a wonderful talk and a very clear description of molecular biology for the layperson. Appreciate that. There are a couple of questions in the Q&A part. One of them from Hertwig is, can patients after injection of the drug via AAV, and by drug I presume they mean the hairpin, of the drug via AAV, participate later in other drug trials? If not, are participants told before they sign in?
That's an excellent question. And every study is going to have its own inclusion-exclusion criteria, so we can't make broad stroke answers to a question like that. It is a standard, exclusion for most experimental therapeutic trials, so most clinical trials, that if you're actively on another experimental trial or another experimental agent, that you can't be in those studies. So that is a fairly standard exclusion. It may not be exclusion for every study in the future. So specifically with an AAV, it's impossible to predict the future, but it is highly possible that if they have an active AAV that's producing an experimental therapeutic, they may not be eligible for subsequent studies. Is that disclosed before people sign up? I can't speak to the specifics. I am not a clinical trial site. I am an advisor in that. Certainly, when people sign consent forms need to include relevant information. It is reasonable to give that information, but again, who's to say that future trials will exclude people that have had AAVs? It will be a trial by trial decision. That said, it's a good point, and people need to recognize that if you're on active trial in one study, you shouldn't, or in most cases, can't participate in new studies.
No.
Most studies will have a washout. The problem with an irreversible treatment like an AAV is there is no such thing as a washout, and once it's there, it's there. So an important point, but again, it's not, we can't give a simple answer that will address every possible future trial. One thing I would say, and it's a bit of an extension of that, is that if and hopefully very soon, when we get to a point that we have an effective therapy, it is likely that much like cancer therapies, we will need to develop our future trials based on best medical care. And if the best medical care is a proven therapy, we now will no longer necessarily have people in the placebo group being untreated, but people in the placebo group will be on best medical therapy versus best medical therapy plus, probably.
Yeah. So another question from Hertwig that I'm going to have to ask you to translate for the audience. What does it mean that NFL does not go down in the OLE study of RG6042? So maybe you could reread that with the acronyms spelled out.
Sure. So I presented data that was in the public domain, and there has been presentations of additional data, and I've given that in some cases. When we, in the original Ionis OLE study that was published in the New England Journal, we looked at a wide number of measurements. We looked at clinical measurements, we looked at MRI, we looked at mutant Huntington in the spinal fluid, and we also looked at other what we call biomarkers, one of which is something called NFL, which is neurofilament light chain. And that's a marker that we can measure in the CSF. There are others like tau that comes from the brain. So in a number of diseases where there is brain injury, these markers go up. And we have done previous studies in the cerebral spinal fluid of HD patients where neurofilament is elevated. Now, nobody has yet done a study, in fact, we're in the process of doing this, where we've taken Huntington's models and knocked down mutant Huntington and said, what is the effect on neurofilament? When we looked at neurofilament in the Ionis Roche study, the study that was published in the New England Journal, there was a rise in neurofilament in the CSF that correlated with the amount of mutant Huntington going down. We have now looked at that over a much longer period in open label. So the first thing, there was no correlation between that rise and any safety concerns. So we don't know what that meant. What we do know is that if patients went off the ASO, the levels came down. When we put them back on it, it went up. And as we continue to treat with ASO, it does go down. So the question actually is incorrect. Neurofilament does go down with prolonged treatment of the specific antisense oligonucleotide that's in the open label trials, tominersen. And it went down to different degrees depending on which of the open label doses was looked at. But when we look at the dose that's gone in now in phase 3 trial, we see this rise, and we don't know what that rise means, and we see it come back down to normal. So if it is a safety concern, it's not correlated with any adverse findings. It's not correlated with any other measure that we know of that would make us suspect that it's harmful. And as we keep giving more of the drug and the drug has its prolonged effects, the levels go down. There are a number of hypotheses, any of which could be true, and some of which we're testing in animals. It could be that the inclusions of Huntington, so as people may have heard, mutant Huntington, one of the things it does in cells is it's quite sticky. It forms inclusions or clumps. Those clumps can be broken up, and maybe the contents of those are released, and one of the things that could be released is neurofilament. It could change the production of neurofilament. Healthier cells make more neurofilament. You make more neurofilament, maybe there's more in the CSF. It could be a sign that some cells are being injured. Absolutely possible. We don't know the answer. But what we do know is that with continued treatment, it goes down. And it does seem to correlate with the level of Huntington lowering. So it's a good question. It's a question that's actively being pursued. But I think from a safety point of view, it's not one that's concerning, not to me.
Okay. And there's, this is probably the most difficult question. What exactly is the role of the Huntington protein in brain cells?
And that's from John Odell. John, great question. One that I've spent much of my career studying in the lab. We've done a lot of studies of what Huntington does. And unfortunately, we don't know a lot, but we're learning. One of the most important things that we do know about Huntington is it's very important in embryonic development. We all have copies of the Huntington gene. We all have 2 copies of it. And there are some people who are born with 1 copy that doesn't work. And it looks like 50% of the normal level, so 1 copy instead of 2, is enough to have normal development. And those individuals are not found to have any abnormalities. So it doesn't look like we need 2 copies. And that's one of the reasons that we say that we target to 50%, because even when it's knocked out throughout life in a human, 50% levels seem to be consistent with normal. In an animal, in mice, if you knock it out completely, they're not even born. So you need some Huntington to be born. The question is how much. If you have 50% the levels, the animals are born and develop relatively normally, although they do seem smaller. So in mice, 50% of Huntington isn't consistent with being completely normal, whereas at least in the humans that have been studied, it does. So what does it do? It's involved in brain cells being born and migrating and developing normally in the brain. After development, after the brain is fully formed, we don't know. There's some evidence that when you have more of it, your brain may be more protected against injury. So it may play a role in brain protection. It also may be involved in different forms of transport within the cell. But at least in animal studies, when it's knocked out in the adult, in the brain, to the levels of even greater than 50%, it doesn't seem to have obvious measurable effects. So it's a great question. It's one we need to continue to study. I think more basic science funding, if it, it unfortunately is very hard to get funding to study the normal function of the Huntington protein. You would think, given how important this is, that the funding would be available, but a lot of the funding agencies no longer are funding studies of normal function of Huntington. So it's a very important question. There have been some discoveries in that space, but we still don't know a lot.
Blair, I wondered if you could make a quick comment on a response from Hertwig about your points about the NFL, the neurofilament. He says neurofilament goes down to pre-drug level, not down to normal. Data presented by Roche Tabriz, and then we'll move on to, if you could comment on that, and then we'll move on to the next.
Yeah, I would love to have also more patient and family questions, but I'm happy to answer Dr. Lange's question as well. So in data that has been presented, but I didn't present, neurofilament does have this elevation, and it does go back to the previous levels. That's the result. I'm not sure what the question.
I think it's saying it
goes to the level of
drug level, but not down to normal.
Normal meaning individuals that don't have HD.
I think so. Yeah.
That would be fantastic, and maybe that's a goal that we hope to get to.
Yeah.
But the result is the result. I'm not sure.
Yeah. So I don't know if you can see this next question from Kimia, and I'll read it so that other people can see it. I'm assuming that gene silencing approaches are most effective for patients during the early stages of the disease. when there is no significant neurodegeneration in the striatum yet. Patients in the advanced stage of the disease may not strongly benefit from them. Would adjuvant therapeutic approaches be more beneficial for patients in the advanced stage of the disease?
I think it's a very important question, and it raises this point that I tried to make at the end, which is we need to continue to develop therapies targeting everything we can. Regenerative medicine is going to be hugely important in the future. Stem cell, targeting inflammation and other approaches. So yes, we absolutely need to continue to develop additional therapeutic approaches. I would argue that any therapy is probably going to be effective or more effective if we can treat sooner. We have a huge advantage in our particular disease in that we have predictive testing and we can determine who carries the causative gene. And we've mentioned some, Leslie mentioned some of the trials, Track HD, Predict. The field has been preparing for pre-manifest Huntington trials for a long time. And we are very anxious to bring trials as early as possible. But they have to be done carefully and they have to be done safely. And we need to ensure that our therapies have a very good basis for efficacy and they're safe. We don't want to be administering or giving a therapy to someone prior to their onset of disease that might have risks. So really, we have to do proper trials. We have to do them carefully. But absolutely, we're super, we have been preparing to do earlier and earlier stage trials, and it's something the field is very anxious to do. But again, we need to be careful. We need to be sure they're safe, and ideally, we want to see efficacy in affected patients first before we go to pre-manifest. And most trials are targeting very early stages of affected individuals, so that's considered sort of the sweet spot for clinical trials. But I can guarantee you, we're all very keen to move as early, and certainly with genetic therapies, the earlier the better. It makes perfect sense. You want to preserve as much brain function as possible, but by all means, we need to continue on all cylinders with other approaches. And whether they're going to be more beneficial in the advanced stages or not, time will tell. But I would say we want to develop therapies for all stages of Huntington's disease.
Could I take the chair's prerogative in asking you the final question? And before we wrap up, it was a great talk. You talked about various different approaches to treating Huntington's disease, but I wonder, as a neuroscientist, you could say a little bit about what you think the impact of these various trials will be for other diseases like Parkinson's, Alzheimer's, and so on, and the notion that rising water raises all boats.
I think that's a great question, and I do believe that the work we're doing in Huntington's disease is going to be applicable to these other more common, in some cases, sporadic disorders, in some cases where there is a genetic basis, but it's not quite as clear. We will be paving the way, and the antisense therapy is there are ASOs that are being developed for Parkinson's, for ALS, for targets that might be efficacious in even more common disorders like Alzheimer's disease and other forms of dementia. So absolutely, every bit of progress that we make, I believe, advances all these neurodegenerative therapies. And similarly with stem cell therapies, if we can develop stem cell therapies and bring them to HD, because of the advantages we have with our wonderful clinical trial groups, our incredible basic scientists, and our established patients who are keen to be involved in our trials, those advances will be applicable to these other disorders. So it's absolutely critical. And in many cases, I talk both to funding agencies and to large pharmaceutical companies. And 10 years ago, we were convincing people to come into Huntington's disease. As you can tell by the trials that are ongoing now, they don't need to be convinced anymore. They recognize that this is an area where we're making advances and they can make advances. And part of that reason they're being involved is that will be translated for other diseases in the future.
Okay, so I'm taking my prerogative for a second last question. You talk about the pharmaceutical companies and I think they're often portrayed as the bad guys just out to make money. But would you mind saying, and I think CERM has realized this, that you need to get biotech and pharma involved in research, and could you maybe say a bit about why that's so important?
Yeah, I think it's a fundamental aspect that I think many of us who are physicians taking care of patients, basic scientists who are developing new therapies, we have to address and we have to deal with. And the translation of an idea into a drug, as you've seen with the timeline I've given, is a very long and incredibly expensive process. And it would be fantastic if it wasn't such a difficult process and it wasn't so long, but we have to do it carefully, we have to do it properly, and we have to do it safely. And that means it's going to be expensive for the near future. And that requires the kind of money that currently, the way the system is would involve companies. And let's face it, companies have the best drug that a company can develop is the safest and most efficacious one. So there is a bias for them to do the best possible science as well. And when the system works well, everything is in concert. I can guarantee you that the academics and the clinicians that are involved in these trials do our utmost to ensure that things are done absolutely correctly. The FDA, the clinical ethics boards are all involved to ensure that we get the right answers. And at the end of the day, we all want the right answers. The companies, the patients, the clinicians, the basic scientists, we want effective therapies that are safe. And that's going to be the most cost-effective and most beneficial for the companies too. So people in the general public who feel that there are conflicts, they're right. There are conflicts. We don't ignore them. We manage them and we address them head on. And that's how we have to deal with this. We have to recognize that companies want to make money, but we have to have a system in which they make money when they do the right thing.
Yeah.
And just to put in a plug here, effective regulations are what are required, not no regulations. So this is a good example of where if the government steps away from its regulatory role, that would be a very bad thing.
Well, that's a subject for another 2 hours of debate. Blair, thank you so much for joining us from Canada. If you'd come to Southern California, you would have just seen smoky skies and maybe felt an earthquake. So we do appreciate it. Very stimulating talk, very clear to understand, and very hopeful. So thank you ever so much.
You're a fantastic group, and I loved the opportunity to chat. And anytime Leslie says jump, I jump. So I'm happy to
copy that. So I'm going to hand it back to our Vice President, Ken, and thank you all for a fantastic couple of hours of science and hope.
Thank you, everyone.
Okay, thank you very much, Peter. You did a great job, as usual, being the moderator. We're lucky to have an academic who possesses such great stage presence. I was thinking as the day went on that maybe the next presidential debate could use you to keep things going a little bit more smoothly.
No, thank you.
I want to offer the board's thanks to all of our speakers today. I thought they were all fantastic presentations. We really covered a panoply of different kinds of topics. I thought that each and every presentation really shined light on topics that are of interest to the entire HD community. And I think we had some topics that were really not just mainstream. So I thought it was a really good set of presentations. I want to thank Dr. Levitt in particular. It was a great presentation, Brian. I think you really did a fantastic job. You and Leslie are clearly Zoom rockstar researchers. And what we have done, the HD Care board, I don't know whether this will show up or not. We have probably not. We have a paperweight heart that's been engraved. We have one for Dr. Levitt and one for Dr. Thompson. And it says, we want to thank Drs. Levitt and Thompson. Oops, I'm sorry. The engraving says, HD Care UC Irvine, in appreciation of Blair R. Levitt, MDCM, and Leslie M. Thompson, PhD, with heartfelt gratitude for your dedication to the Huntington's disease community, October 3rd, 2020. And I really want to echo that. I think that those of us who have had HD affect our families really are blessed to have people like Blair and Leslie dedicating so much energy and such a great part of their life to doing research for HD Care. I mean, for HD defeat. HD Care got really top-notch support from UCI Media Organization. I think this whole day came off really well. HD Care was stepping off on an endeavor that we had no experience with. We've never done a webinar. We've never done virtual like this. And we were doing it with very little time for preparation. So the hand-holding that we got from UCI Media really was a help and very much appreciated. Finally, it is not a so well-kept secret that tomorrow is the birthday of our founder and inspiration, Frances Saldana, the HD Care President Emeritus. As we all know, and as was mentioned earlier this day, she has just really been an inspiration for the HD fight for many years, really many decades, and across the country. So I ask everyone to offer up a not so virtual round of applause to the person who keeps our mission alive, Frances Saldana. Okay, with that, I have nothing else to add. If no one else does, then we'll call this symposium adjourned. Thank you very much.